Hello, I'm Mike Buschmann. I'm going to give just a short presentation on some potential research projects for new graduate students. I've been at Mason since 2017. Prior to that, I was in Montreal for several years, and my general area of research is in biomaterials and nanomedicine. So the research that I do at Mason is in the area of mRNA therapeutics. So mRNA is the intermediary between the gene and the protein. And as you can imagine, it can be used anywhere as a therapeutic where you would use a protein. So one area is in vaccines for infectious disease, such as COVID-19. That's indeed our main project currently. And the mRNA can produce an immunogen, so a protein antigen, namely the spike protein in that case, and then activate an immune response that hopefully protects vaccinated patients from being infected by that virus. The other area that's very active and advanced is in cancer. Generally, the same theme applies, except you activate the immune system against a tumor and remove the tumor in that means. And then an additional area is in regenerative medicine, where you can also produce, for example, a growth factor, and a growth factor that could stimulate vascularization. In this example would be for after a heart attack for ischemic heart tissue and repairing that. So the problem that we address is in the delivery of these long single-stranded nucleic acids, the mRNA, typically thousands of nucleotides in a row. And we do that by packaging them inside these lipid nanoparticles. Typically, there's about 4 lipids that produce the nanoparticle, and then the 5th component is the RNA. Here it's actually a silencing RNA, since that was developed over a longer period of time. The way they're manufactured is through a microfluidic system. So the lipids are injected in one channel in an ethanol solution where they're soluble, and the RNA is in an aqueous channel that's meeting the ethanol solution with the lipids, and the lipids become less soluble because they see water, and they also ionize. So the cationic lipid will become positively charged and bind the negatively charged phosphate backbone and get mixed up in these mixing element components. And then in a couple of milliseconds, the particles are formed spontaneously, they come out the other end. And this whole thing is held together through that positive-negative electrostatic attraction between the positively charged ionizable, the negatively charged nucleic acid, and then hydrophobic interactions that form the surface with this hydrophilic pegylated lipid on the outside. So once you have these particles, if you inject them into cell cultures or into animals or people, they will eventually find a cell surface and get internalized. That's the first barrier that has to be overcome. But they get internalized into an endosome, and an endosome is a little vesicle in itself, and the payload has to get out of the nanoparticle, out of this endosomal membrane here. And the way it does it is the pH will start to go down in the endosome below 7, and then the ionizable lipid has to become positively charged. So you can see right away it has to have a pKa that's maybe 6.5. And indeed, that's one of the main design criteria of these lipids, is they should have a pKa in the LNP lipid phase that's about 6.5, because then they form these ion pairs after they become protonated at maybe pH 6. And those ion pairs then lyse the membrane and allow the payload to go out. And in the case of mRNA, it would be in the cytosol and able to find a ribosome and then be translated into a protein. So the efficiency of this endosomal rupture mechanism that depends on ion pair formation is actually quite weak, around 2%. So there's a lot of effort, including our lab, to increase that to maybe 20%, in which case you could have highly potent systems and reduce any potential side effects. So since charge is so central to this whole delivery issue, we look at the ionization of the nitrogens on the potential ionizable lipids that we can use. These are published ones. I can't yet show the ones that we've actually synthesized and created ourselves, but we analyze those similarly, and we look for what is the pKa using some theoretical calculations. And if the pKa is sort of in this range here, we expect once they get into the lipid phase, the pKa will be about 2.5 points lower, which is an interesting phenomena that requires more time to explain, but we know that this range is good for the molecule because of that 2-point drop. And so we look for these kinds of pKa's in the new lipids we're synthesizing as the first design criteria for them to be successful. And we also characterize the pK of the synthesized molecule using NMR. If you follow the chemical shift of the spectra, it changes just like the Henderson-Hasselbalch curve predicts. And you can measure the pKa and be very sure that your predictions are accurate, so that you can design by theory and not have to synthesize and test every single candidate, which would really take a long time. Then we characterize the ionization in the particle itself. We do that with a method called zeta potential. So we put the particles in an electric field, see how fast they move. And if they move in one direction, they're positive. If they move in the other direction, of course, they're negative. So you get the zeta potential from that movement. And at low pH, the ionizable lipid is entirely protonated, so they're highly positively charged. And then as pH goes up, they become neutral, they cross this line, and now they're negatively charged at high pH, where you only have the nucleic acid negative charge remaining. So we look very carefully at this curve. We understand how it depends, for example, on these formulation parameters and on the choice of the ionizable lipid. And we can relate that to where these particles traffic. Positively charged particles go to some locations, negative to others, neutral yet to others. And then once they're there, if they ionize properly, they can be out of the endosome and then translated on the ribosome to produce the protein, which is the final product that you're looking for in people or in cells. We also use standard techniques to compare these ionization through the pK from this technique or this one. We compare them and make sure we understand the ionization from all points of view. And then we also have molecular models, mathematical models that we use to calculate things like how many copies of the mRNA are in each particle, which is a very important parameter also to know how effective they can be. Finally, we put them in cultures. This is an example of the expression of a protein at different doses compared to the commercial product that's already approved. And we have fairly similar expression for most doses for the first family that we synthesized. We also look at their ultrastructure. This is very important to see using a cryo-electron microscope if they're homogeneous in nature or if they're kind of segmented or compartmentalized. Those have very important influences on their function. And then we inject them into animals. And we found recently a very interesting phenomena with some potential vaccine candidates where our newly synthesized lipids are retained at the site of injection and the draining lymph node from the muscle. And the commercial systems actually seem to go all over the body. So you inject them in the muscle and you find them 4 hours later in the liver, for example. And this happens very commonly for most, in fact, all commercial systems we've tested to date. So we believe the new family that we have here may limit systemic toxicity and adverse events since they are really retained locally. And in fact, those systemic and toxicity and adverse events are issues in the recent COVID-19 trials that are very relevant and possibly improved by some of these discoveries we've made. So we're looking at the immunogenicity of our LNPs delivering a SARS-CoV-2 immunogen for the spike protein. And then we're also synthesizing the next generation, which we expect to be even more efficient than these. And so for projects for new graduate students, most of them revolve around the vaccine area. For graduate students who maybe have a strong chemistry background, that would be the design, synthesis, and characterization of the lipids. For engineering backgrounds, there's a lot of things to look at in the microfluidics, mixing regime, modeling, ionization events, and physical chemical events and engineering questions that still remain to be solved. And then there's the whole area of the electrokinetics, ionization, charge-dependent delivery that we look at both experimentally and theoretically. And these same systems are, of course, applied in different areas. One of them is very interesting, is that when you take an mRNA LNP system and a cell will endocytose that system, some of it can be released and translated on the ribosome, but some is actually repackaged in another vesicle called an exosome and is exported from that cell. And so it can go somewhere else in the body. Could do something good, could do something bad. And we kind of want to look at what's happening with these exosomes as well. So that's a new project we're just starting. And the last one is in gene editing. If you have 3 components in the lipid nanoparticle, an mRNA Cas9 that cuts the genome at a certain location, guided by a short RNA sequence called the guide RNA. And if a third component, the plasmid DNA, is there with a gene sequence that you want to insert, you have the ability to delete and insert a new gene at a specified location. And again, delivery issues are critical here. You have to be able to package these in a very defined manner inside the LNP and have them all be efficiently delivered in order to succeed. So if you're interested in any of those projects, contact me please by email and we can discuss further, perhaps on a phone call or a Zoom call. Looking forward to talking with you. Thank you.